Not a bad idea. If you give me maybe an hour after class, I should be able to have homework for ready to go. Yeah. For sure the due date's been pushed. So where we ended yesterday was looking at the acid-base characteristics of amino acids. So you should be working on getting some memorized and understanding something about the basic structure within your amino acids. So there's all amino acids have two primary functionalities. We have the amine, so your NH2. Okay, so you've got that amine portion of it, of an amino acid, and then we also have the carboxylic acid portion of it. And you'll notice really at no point do we ever end up with just an amine or just a carboxylic acid, or the amine and the carboxylic acid functional group at the exact same time. Okay, why not? What's the reactivity of a carboxylic acid?
2.6 or something.
Kind of. That's quantifying it. Give me less quantification. What's the reactivity? How would it act? It acts as an acid. How do amines act? As bases. So anytime you put an amine functionality and a carboxylic acid functionality in the same molecule, carboxylic acid is deprotonated by the amine. Okay? So you get this kind of weird dynamic because you have both a base functionality and an acid functionality in the same molecule. Okay? So you do get some kind of odd charges showing up within those, which is one of the things that makes amino acid memorization a little bit tricky, because depending on the pH of your system, you're going to have a slightly different structure. What's the only thing changing when you change the pH, however? Just the hydrogens on the structure. Okay, so there's a variety of ways that you can go through and try and memorize those amino acids. The flashcards, which I did end up getting posted, I think, to useful files, those are shown at physiological pH. Okay? From a former student that has taken the equivalent of this class at other community colleges, says the easiest way to memorize the amino acids is just memorize the physiological pH. Ultimately, there's at least 3 different structures. It doesn't matter which one you memorize as long as you realize you can go different directions off of it. I personally think memorizing the physiological pH one is the most difficult, because I now have to remember that I can add hydrogens and remove hydrogens. Okay? Personally, where I would probably start is going to an extremely low pH. Okay? What happens at a very low pH? Okay, we've got tons of hydrogen ions in that solution. So at such a low pH, there's so much hydrogen ions that anything that can remotely pick up a hydrogen ion is going to have a hydrogen ion attached to it. So when we go through and take a look at the functionality of our amino acid, we've got the carboxylic acid functional group, which will have a hydrogen attached to it, because our pH is lower than the pKa of that functional group. If we go through and take a look at the amino functionality, you'll notice that it has picked up an extra hydrogen as well. And that's again because the pH is so low, it's much lower than the pKa of that functional group, that the hydrogen stays on. Okay, so if you remember back to our acid-base chapter, we did a comparison between the pH and our pKa. If the pH was less than the pKa, what did that mean? We're in our hydrogen added structure, so we would call it HA if we're going to go generic. So H for our hydrogen and A for acid, but it could really mean anything. If we looked at the case where the pH was greater than the pKa, what does that mean? We remove the hydrogen, and the structure we're looking at is our A minus. Okay, we got to be a little bit careful with charges, but what we're looking at is either having the hydrogen on or the hydrogen off, depending on the relationship between the pH and the pKa. So how do you go through and interpret that with the pKa information? Well, you'd have to know your pKa's. Okay? So there's a couple of them that you absolutely should have memorized. Okay? And that couple is really literally a couple, just 2. The 2 that you really need to have memorized are the pKa for the carboxylic acid of the amino acid, which is roughly 2. Okay, think I've got the exact average on there somewhere. It's roughly, I think 2.2, but 2 is close enough. And the pKa for the amine, we're looking at roughly 9. So if we go back and take our central comparisons, if we're at an extremely low pH, pH less than 1, our pH is lower than the carboxylic acid, which means I should have the hydrogen on my structure. And sure enough, at a low pH, we've got the hydrogen on the carboxylic acid functional group. The next part is to take a look at the ammonium. pKa less than 1, it is still lower than 9, which means that functional group needs to have that hydrogen. So we have NH3 plus. Why is it not neutral? How did nitrogen act? As a base, whereas our carboxylic acid acted as an acid typically, so they pick up opposite charges. Your acid will typically pick up either neutral or negative based on that hydrogen. Your amino group will either pick up the positive or neutral, depending on its pH. Okay? What I would then do is jump to the extreme. Let's go all the way up to a wicked high pH. Could drop a little bit lower than that 12, because ultimately it's in comparison to these two right now. Any guesses why I went all the way up to 12? Okay. To exceed all doubt, and that all doubt is really going to come back to the R group. You'll notice with the structures we've got up here, we haven't specified what that R group is. One of the amino acids has a pKa up right around 12. Okay? So I'm trying to really clear that limit, make sure we aren't even concerned about the R group. Okay? So if we're up at that high pH, we again can go through and compare. Carboxylic acid has a pKa of 2.2. Our pH is much greater than the pKa, which means I should be in the non-hydrogenated state. In the case of my acid, I pick up the negative charge. We then evaluate the next functionality within our amino acid. We're looking at the amine part. The pKa of our amino acid is 9. In comparison to 12, it's less. If it's less than, our pH is greater than our pKa. Got to make sure we reference that the same way. Again, we want to remove the hydrogen. Remember, we're not removing an extra hydrogen from our NH2. We're removing it from our acidified position. So it drops to the NH2 from the NH3. Kind of, sort of? Okay. So what happens in the middle? So now I'm going to have to erase everything in our middle. So we lose that little key. What happens if we're somewhere in between? So we're ultimately looking at, in this case, a pH somewhere between 2 and 9. So let's put it up at physiological pH, pH 7. And if we draw out our structure, make sure I draw the correct arrangement. I'm just going to do NH2 for the moment, and we'll do COO for the moment as well. And we'll then go through and decide. Our carboxylic acid, pKa of 2.2, pH of 7. The pH is greater than the pKa, which means I need to leave the hydrogen off. So I will not add that hydrogen to the COO. What charge does it need to be? Negative. Now we can go through and look at our other functionality, our nitrogen. Okay. pH of 7, pKa versus 9. Our pH is less than our pKa, which means the hydrogen needs to go on. So now I can go back to my NH2, and I need to add that extra hydrogen. So we could just go through and do it this way. Our nitrogen was neutral to begin with, so now that needs to become positive. Okay. The interesting thing about this is that we now have what's known as the zwitterion. We have an overall neutral structure, but it's charged. It has a positive and negative. Okay. This particular species will also carry an interesting name. Not so much the zwitterion aspect, but we're really concerned about this structure itself. And it comes up with another name. We're looking at what's known as the isoelectric point. Okay. the overall charge is neutral on the molecule. Just so happens in this case it's also a zwitterion. Right? So if we have that isoelectric structure, there's a lot of, and I'm not quite sure exactly why, there's a lot of kind of explanations around that. So you'll hear reference to that point. Right? In particular, it's usually referred to as the PI. Right? It's referred to as the PI. So when you're looking at a titration curve, if you want to find your isoelectric point, they're really referring to the PI on your titration curve.
So the PI would be 7 as well?
So we'll have to be a little bit careful about that. We'll need to look at an exact structure to nail that down. But yeah, we'll look into that. Ultimately, your PI is dependent on the 2 pKa's that bounce around it. Okay? So as we're shifting from one situation to the next, the PI is the dead center middle. Because any variation up or down from that, we will have either started removing a hydrogen, in which case we're not at the exact isoelectric point, or we would have put on a hydrogen, in which case, again, we're not at the exact isoelectric point. So your isoelectric point is the exact point that you have the plus and the minus in equal concentrations.
If the R group has a pKa, is that correct?
So the R group will absolutely have an impact, because if your R group is charged, okay, let's say at this pH, our R group carries a negative charge. Are we at the isoelectric point? No, because now our molecule is overall negative charge. Okay, so your isoelectric point is really dependent on the overall structure, including the R groups. You need to make sure you have a neutral overall charge.
So does that mean you have to add towards the PI?
We'll look at more specific examples of dealing with the PI when we really get there. I don't know what the P is, other than when we look at the PI, we're getting it from the pKa and the pH. So that's where the P is ultimately coming from. The I stands for isoelectric. Okay? So this is what was supposed to stepwise, and in theory we'll stepwise now. Okay, so again, like I said, there's varying ways that you could go through and look at your individual amino acids. I would almost prefer, to go to the complete acidic form, which means we need to have that hydrogen on there. Okay. In that completely acidified state, now all I have to do is slowly remove hydrogens. Okay. I don't have to worry about what the physiological pH is, because I can find that based on all the other information given through those structures. Okay. So I personally would probably spend time trying to memorize the fully acidic form. And if you take a look at all the examples that you see in your textbook, when we look at titration curves, we're usually starting with the acidic form and we add base to it. Okay? There are going to be kind of some exceptions to that. Would we expect to find the acidic form in the body? Why not? Our body, we're looking at pH 7, so some of that chemistry becomes a little bit weird, so it's ultimately going to come down to what ends up being the most useful for you. Do you want to know the structure to be able to work with it, or do you want to know the functionality of when it's going to be processed in the body? Okay. So if we start with our fully acidified structure, okay, first thing we should go through and do is evaluate all the possible reactivities on this structure. So where do you see reactivity? Carboxylic acid is one, which is nicely labeled with our hydrogen. We've got the amine. I'm going to make that an NH2 and accent that hydrogen coming off. And then we also have that hydrogen. And I really don't like you referencing that as an alcohol, because it's not an alcohol. What is it? It's a phenol. Why do we refer to it as a phenol and not an alcohol? The location of that hydroxy group next to the aromatic ring drastically changes the reactivity of the hydrogen. The hydrogen now becomes acidic, whereas the alcohol has virtually no acidic characteristics. Okay? So we have 3 functional groups here. So this is a step up from what we just looked at. So what we're going to try and do is stepwise through, in case you couldn't guess, we're going to look at a couple different structures coming off of this. So between those three acidic hydrogens, which one's the most acidic? The carboxylic acid. Since it's coming off of our amino group, that pKa we roughly know to be 2.2. So we could step away, we're looking at 2.2. What would our structure look like?
That one.
Shoot. That one, I'm going to have to keep drawing a hydrogen in there. Okay. All we're going to do is remove that one hydrogen, okay, from it being the most acidic. Okay. Again, we want to stepwise through. So what's our next most acidic functional group? Okay. That becomes a bit tricky to figure that one out. Ultimately, it's something that you would look up. And as Braden is very nicely proving for us with the textbook open to the list of pKa's, the pKa of our amine comes out to be roughly 9. The pKa of a phenol, 10. So which one's more acidic? The amine is more acidic. So if we're going to increase our pH a little bit further, eventually we're going to end up losing the hydrogen off our amine, being our 9.11, and our structure now looks like that guy. Keep going back and adding that stupid hydrogen. Okay. We still have functionality in that R group, so what can happen now? We're going to lose that hydrogen, so we can look at the pKa for our phenol being at about 10, and we can remove that hydrogen now. Okay? So, draw that other hydrogen again back on there. Copy paste. So the flashcards that I put up are at pH 7, physiological pH. Okay? So what we would want to go through and do now is take a look at the charge for each of these structures. So what's the charge on the very first structure? It's positive. Charge on the second structure. Third structure. Fourth structure. -2. Where's our isoelectric point, or our isoelectric compound? Our neutral one. So this is our isoelectric. Clearly someone has read ahead a little bit. What is the pi? So it's the point at which we have purely this compound. Okay. So it's going to be whatever the pH is when this hits, but this hits halfway between these two structures. Okay. So the exact halfway point is going to be the average of these two pKa's. Okay? So when we look at calculating the pi, what we're effectively doing, oh, I didn't give myself a lot of space there.
So halfway between the pi is also equal to
The pH at that point,
That's
yeah.
what I'm saying, at that point.
Yeah. So our pi in this case will be 9.11 plus our 2.2 divided by 2. And we now have the pi. Okay. One thing you've got to be careful of, is it always going to be the first two pKa's? No, because depending on that R group characteristics, we could be using the last two. Okay. Histidine, I believe, is one that's going to do that to you. Okay. How do you decide which two pKa's to use? Either side of the isoelectric point, but how do you know where the isoelectric point is? It has neutral charge. How do you know it has a neutral charge? You would draw it all out. Okay. Feel free to look up. There are other little formulas. I think your textbook mentions you can calculate the pi for things that have R groups that are odd, okay, or that are reactive. And they go through and say if it's acidic, then you use the first two. If it's basic, you use the last two. I can't consistently get that formula to work. So I'll tell you right now the way I would calculate the pi, I would draw out every single structure, put in all of my pKa's, identify the charges. Wherever my neutral state is, I'm going to look at the pKa's that surround that neutral state, and that's what I will average. Right? If you find a textbook reference, and it doesn't have to be your textbook because I don't think yours explains it very well, that actually sets it up so you can say how to calculate the PI, great, let me know and we can share that with the rest of the class. Every method that I've seen, it's probably easier just to memorize the PIs on top of all the pKa's. Okay. When is this PI issue going to become a problem? Only when we have 3 pKa's. Okay, and the homework will hit you with it a couple times, but really the only time we're concerned about the PI being problematic is with 3 pKa's. Because if we only have 2, oh, I don't want to stepwise, never mind. We went back to that previous slide. If we only have 2 functionalities, the average has to be those 2. We don't have a choice. So we're really only looking at the R groups, or looking for weird PIs where our R groups have some functionality. How many R groups have that extra functionality? 5 or 6. So you could go through and memorize some method to go through and solve, but to me the amount of effort to memorize that method is probably more than just memorizing the PIs for those 6. Okay? That's why I would draw out the structures, because then I'm already memorized the structures, I can apply from that. Okay? As far as the exam goes, what I would expect on the exam, if I asked you to calculate a PI for a particular amino acid, okay, I'm not going to assume that you've memorized the pKa's. What I would list for you is, here are the pKa's for this. And it won't necessarily be in sequence. Okay? You should be able to match them up. So if I gave you a pKa of 4, 2, and 9, the pKa of 4 applied to what part? Your amino, your carboxylic acid, or the R group?
R group.
Has to be the R group. Why?
Because 2 is not
2 and 9 are your acid and your amino. Okay? So be aware of that also when looking at the tables that you're given. When they list the tables in the textbook, it's usually acid, amino, R group. Those do not necessarily flow from lowest to highest pKa's. Okay, so you got to watch out for that sequencing. Read your tables carefully. So I see a hand, and I'll come back to you. Yeah.
In that exam, so would we be recalling the structure? I mean, you're giving us the pKa, but we're recalling the structure that we would have memorized.
I would probably give you a starting structure as well.
Okay.
Okay. When it comes to the amino acids I expect you to memorize, that's probably going to be a very quick, give me 3 amino acids. Any question thereafter that I would ask about something about the structures, I would give you the structure. I wouldn't tell you what the amino acid was. So that you couldn't go to that section of the test, go, oh, okay, that's histidine. Oh, okay, I'm going to draw histidine, and that's histidine, and HIS, and all that stuff. Okay. You can probably get away with a little bit of that, but it's not going to get you full credit. Okay? Yes?
The pKa is the 2, 4, and 9. You average the 4 and the 9 probably.
I think I'd have to draw it out. I really am that bad with it. I would have to draw it out to see where those charges showed up.
It's tough.
Yeah. I just don't see a pattern there. Structures are my bread and butter. I'll deal with structures any day. You give me some formula, it's going to confuse the hell out of me.
You were talking about memorizing. You said you wanted to do that. 1 letter code in the structure.
Yep.
Not pKa's.
You don't need to worry about the memorization of pKa's.
Okay.
Within reason. The acid and the base, you need those.
Right.
2 and 9. Okay. Other questions on this? Okay. Just like we did with our acid and base chapter and buffers, we're going to have to go through and be able to interpret titration curves. So if we take a look at tyrosine, does this titration curve match tyrosine?
No.
Why not?
As 2.
Tyrosine has 3 pKa's, which means there's 3 points at which it can buffer, which means there should be 3 points on this curve that, how do you know it's a buffer? You get a horizontal section in your curve. Remember, the horizontal section means that the pH doesn't change when you add that base. Okay? This curve only shows 2, which means Can't be right. Okay, so let's draw one. Where are we going to start? On the acidic side, and what's going to happen? Okay, our carboxylic acid will be the first thing to be deprotonated. Okay, where should the center of our buffer system be? Remember, our buffer is a flat space. The height of that should be at the pKa. Right in the dead center of our buffer needs to be the pKa. Remember, pH equals our pKa at our half equivalence point. So what we could go through and do is start to specify some information here. We look at our tick marks. So for our first buffer system, we would expect a flat line about 2.2. So we're looking maybe somewhere about here. Okay. So we would expect some buffer system to be flatlined around that range. The exact volume of the NaOH, I don't even really care about that. I want to see the basics of where these things fall. Okay. Where would our next buffering system be? I know that's kind of small, so the people up front can help you out. We're looking about 9.1. So our next buffer needs to happen 8, 9. I think I got about 9 there. Pretty close. And then our next buffer, I'm not using that many sig figs. We're looking at about 10. Oh man, that's not ruled very well, is it? 8, 9. Expect something just a little bit higher up. Okay. How are we going to connect these? Okay. What happens outside of that buffer range is we might maintain roughly about a 1 pH unit change either direction, but then it's going to immediately spike. Okay. And yes, the spacing on this isn't perfect, but I'm not concerned about that. We're going to see it kind of level, and then it's going to spike, and then it's going to level, and then it should spike, and then it should level again. Okay. And now we've predicted ultimately what our titration curve should look like. Given the titration curve, how do you find out what your pKa's are? We'll go for those flat spots, find roughly the dead center of our flat spot. The dead center, jump across, look at our pH. We know that to be true from our Henderson-Hasselbalch equation. pH equals our pKa plus our log A minus over HA. I'm just going to do that. Okay. A minus will equal HA at the half point on shifting on our pH. Okay, so at this point, we've got our pKa 1, which should represent the carboxylic acid. We move to the next flat point. I drew that one really flat. We go back across to our pH. At that point, again, we now have our pKa 2. We look at our next flat point, and we'd have our pKa 3.
Wouldn't that be H2A and then HA? And then HA?
Yes, as we shift further up, you're right, those exact terms are going to change. The reason why I won't change it right now is I'm lazy, but we can kind of get away with that because what we're referencing is the loss of a single hydrogen in each case. Okay. So to transition between here and here, yes, it is H2A, but it's H2A to HA. That's the equivalent of this. Yeah. It's still looking at the conjugate relationships. Okay. Questions about that? In case you couldn't tell, being able to interpret graphs is going to be a pretty massive part of this overall class. That's the way that I feel that we can still cover the material without having to go into the brutal mathematics behind a lot of this. Okay? Questions? Yeah.
So like, because on the homework, there was like a line, what is this? Is that, like when you go up, like the steep, what is that?
Oh, I see what you're saying. So what information can we pull from this? I know what you're referring to, and it's a good thing that we came back to that.
They also did a OH
So,
equivalence, which
yeah, and the OH equivalence is just going to be referring to pulling off a single hydrogen each time. So that you're just looking at the rough amounts, whereas the milliliters is too difficult to interpret. Okay. So if we're looking at, say, that orange point, okay, what is special about that orange point? In the case of tyrosine, it was our PI. If you go all the way back, and it's probably easier to see on the previous slides, if we go through and evaluate where our PI was located, it was our central structure here. It's halfway between these 2 points, those 2 pKa's. Where are those 2 pKa's on our curve? pKa2 and pKa1. Yeah, the whole hand drawing thing, you're going to lose all sorts of issues with scale, but okay, halfway between those 2 points is going to give me the PI. The exact point in which I lose a hydrogen, okay, to get me to that neutral state. So I'm exactly halfway in between those transitions.
So is that typically on the steepest part?
It'll always be on the steepest part. Because remember, how do you calculate the PI? It's the average of your pKa's. It's always going to be dead center on a steep section of your curve.
I was confused about that.
Yeah. So
Yeah.
is that because they can't buffer? Because it's like neutral? That's like kind of what water is.
It's a good question on that.
Because like, I mean, I'll say I just guessed. I mean, like, middle KPI. 'Cause I knew what it was. But I kind of want to know.
almost
You
really
can't buffer at that point. Yeah, actually, I'm not sure that that really matters, but
It doesn't act like a weak.
because you only, well, just because you're neutral doesn't mean you can't be an acid or a base. So that doesn't quite work. And in fact, with 2 charges, you should be able to both be acidic and basic. So you should still have that ability. You're just, yeah, I'm not sure exactly on that.
You know, post it like some on campus.
Yeah, I'll see if I can't push that a little bit more, see if we can get some more information on what's going on with the PI. Whoops, should coat that in orange. So our PI is at that halfway point. You were saying something too?
I was just going to say, to me, I don't try to associate the PI with the pKa's because I'm looking at acid, base, and neutrality as far as the charge goes. So I try
Yeah.
to separate the 2 out. I mean, I know they're all related and associated.
Yeah, they're all
because of
related,
that.
but yeah,
You
you're
Separate
right.
it out, you say, okay, here, this is for charge and this is for pH.
Yeah, that might just be the best way to look at it, that your PI is in reference to charge. So all we're trying to do is find that charge, whereas your pKa's and all the pH stuff is all about looking at acid concentrations, buffering abilities. It's not necessarily about charge. They're related to each other, but if you separate those 2 concepts, then we don't have to think about it as much.
It says that the molecule will not migrate in an electric field, and this property can be put to use in separation methods.
Yeah, and we will take advantage of that in separation. So, and that might be what they're looking for, because if we can adjust the pH to get it to a proper PI, get most of, say, our amino acids to a certain PI situation, then some of them that have higher or lower PIs will carry a charge, and we can then separate those away based on adjusting the pH. So it's sounds like it's going to be primarily a sorting technique than anything else.
So PI is where the greatest change then?
PI will always be at your greatest change. And if you have multiple great changes, so be careful on that. So here we're seeing a change 2. Remember, this isn't to scale.
Right.
Okay. So what you're looking at is wherever you have a sharp change in your slope.
And the charge.
Okay, if it goes vertical, and you have a charge of 0 on your structure. So to find your PI, this is where, again, I would go back to the structures, figure out where it's neutral, and average the pKa's around it.
Okay.
Okay. Once you've got that, you can go in and label where the PI is on your graph.
Will there be any times where they want you just to find the PI and
graph? If there's like multiple.
That's definitely not in the homework, right? I don't think that was in there.
But
I don't remember.
They
Finding
had
the PI. There was something where you had to put the PI in there.
They had a graph. That was
But you have charge, so you do it based on the charge. You don't have to do it based on
I remember that.
the PKA and the
Yeah, the
PA.
final graph kind of, you put everything together. And
Yeah.
You have the charges of the PI and the
Well,
PKA.
And so you could base it with the charge.
You had to label the pKa's of the carboxylic acid mean, and then the PI, they asked me to enter that in afterward. Yeah.
Yes, but that goes back to the structures. And so to answer that question is looking at the structures.
Yeah.
Yeah.
And so you just have to use that equation.
Yeah, so it's not using a titration curve on its own. It's using the titration curve and structure or charge to interpret your PI.
So if you can use the PI as a separation between like the structure of pKa2 and the structure of pKa1, would there be like almost like a PI between pKa2 and pKa3 that you could get the pH2 to separate?
You
those
can
two
take advantage
structures?
of, so careful, when we're separating our structures, what are we separating? We're separating different amino acids. So we're not trying to separate the structures within our amino acid patterns, because once we get our amino acid out, we can put it in any structure we want. We're taking advantage of the charge and ultimately our PI kind of relationships to separate amino acids from each other, not amino acid states
Gotcha.
or an individual amino acid state. Okay?
So if you had two amino acids in one solution, would you have to take both PIs into consideration
Yep.
in order to separate them?
Yep. You would take a look at both their PIs, try and get
Average those.
your well, no, you try and adjust your pH to the point where one of them is located at its PI. It now has zero charge. So now if I put that solution under electric field, the one with zero charge isn't going to see that electric field. And depending on how we've got the experiment set up, it might either not move or it might run really fast, whereas the other one would then be stuck. Okay? Other questions on that? We will do that in lab, by the way. I'm pretty sure we're looking at electrophoresis with our lab. So our isoelectric point, we're looking at no net charge on the molecule. So we use that to kind of take advantage of separation. It's looking at the average of the two pKa's. In the case of most amino acids, so we take a look at this list, this is coming from your textbook, most of them, your PI is just the average of the only two pKa's you've got. So it's the pKa of the acid, the pKa of the base, and we don't have to worry about anything else. Where it becomes tricky is if we have more than two pKa's. Okay? To evaluate that, that's where I would say you go to the structures. Okay? Like I said again, there are formulas that allegedly tell you which two pKa's to use. I just haven't been able to get that to consistently work, so I'm not even going to attempt to do it. The structures I know work. So if we picked, say, tyrosine, which is what three-letter code? TYR. Let's see if I can underline this. This might take way too steady of a hand. We look at our tyrosine here. Which two should we average?
First two.
Okay, well, based on just those numbers, I don't see a pattern that particularly works. So what I would do is go through and take a look at the structure, identify where I have my zero charge, and this is where we have to be careful by first two.
Yeah.
Okay. Our zero charge was this guy. I need the pKa1 and the pKa2, which is in reference to the acid and the base. In this list, you're right, it's the first two on here.
Right.
That may not correlate with the first
No.
two on here. So be careful in your terminology. Okay. So a good example of that one is if we looked at, say, glutamic acid with what symbol? GLU. Right. We just said the first two. On this list, that looks like these two. But if we look at pKa's, it could very well be these two.
So what's the lowest two?
It's, the PI is not always the lowest two. It depends on the functionality. So that's what I said, draw the structure. Okay, and I'm pretty sure with glutamic acid, it's the last two, actually. I think it's the average between the 4.2 and the 9.67.
They have their greatest difference.
That might be an interesting pattern to look at and see if that consistently works. That might work. I would just test it, really. I would just test it. Like I said, I've looked at a few different formulas that claim this is how you calculate the PI, and I don't see it always working. So I'll go back to the structure where I know it works. Questions on the PI? Okay. What kind of questions might you see on the exam on PI? Might very well get a structure like tyrosine. I would probably only provide you with one of these structures, and I'd list the 3 pKa's. And I'd ask, okay, what's your PI? Right? To do that, and I'd probably ask for you to, I don't know if I'd ask for you to show your work. I would expect to see all the structures drawn, the pKa's labeled in, finding your neutral one and taking the average of the 2 pKa's around it. Okay? If you can come up with some fancy formula that works, then hey, you get that answer a little bit faster. Yeah?
If there's 3 pKa's, are you going to tell us if the R group is which pKa belongs to the R group?
No, I won't. So if we go back one slide, you take a look at our R group pKa's. Why would I not tell you what our R groups are? Okay, we know 2 and 9. If you take a look, and it's not perfect, but the first one, almost all of them are 2s. We see a couple high 1s. If we take a look at the amine group, we're looking at relatively high 9s for most of it, maybe 9.5. We see a couple that hit up in that 10 range, which makes it a little bit more difficult. When you look at your R groups, most of them fall outside of those ranges. Okay, so as far as the sake of the exam, I would pick one that definitely, that is very characteristic set out from the 2 and 9. Thank you, Adam, for the head wagging. Okay. Yes. Oh.
Are you always going to give us the most acidic structure, and then we'll have to do it from there, or?
I will probably vary it, but I would guess I'll give you the most acidic. Yeah. Other questions? Okay, isoelectric point. So you can look up tables that list all the pKa's and the isoelectric points that go along with them. So this is how I tested a few different formulas. I looked up what the official PI was and then tried to calculate it based on those other formulas. So there's only, what do we say, 6? 1, 2, 3, 4, 5, 6. We got 7 of them. There's only 7, so you could probably go through and figure out which patterns you want to follow if you really want to do that. Otherwise, go to the structures. The structures will help you out.
Are we going to need to know the first letters, like the 1 letter?
You need to know the 1-letter code, yes. You need to know for 5. For 5, you are required to have them memorized.
Oh, it's any 5.
Yeah, any 5.
Just spell your name with them. That's what I did.
That could work.
Alphabet. I just spelled
It's
my
pretty
name.
close to the alphabet. It's not
Yeah,
perfect.
so I messed with it or worded it.
But there's no Z.
Enough of that. Okay. Here's your pH comparisons again on what's happening between your pH and your pKa. Okay. That is a very important relationship. You are guaranteed to see questions on that as far as determining your protonation states. General basics of your amino acids. You got 20 amino acids. They're referred to as alpha amino acids. What's the alpha mean?
Carbon versus something.
When we draw out our amino acid, here's our acid group, here's our amine group, that's our alpha carbon. What if it was a beta amino acid?
Away from the alpha.
And a beta amino acid. So it's just referring to the carbon sequence. Alpha, beta, what comes next?
Gamma.
Gamma, okay, which is, I shouldn't wave that around. It's kind of a weird looking Y. Okay. 19 of the 20 are primary aminos. So that means when you look at the nitrogen, it has only one carbon attached to it. There is an exception. Proline is a secondary. If you look at the structure of proline, it's very distinct from the others. I'd recommend that that's a good one to memorize, regardless of what you consider already, because that side chain actually doubles back on the amino acid, okay, which is going to restrict a lot of the motion within that amino acid itself. Okay? That restriction in motion can now force structural effects further on down the chain. All right? 19 of the 20, we have stereocenters. Okay? Which one? Oh, man.
Yes.
Okay, let's try it again. Why is glycine the exception?
Because it's hydrogen.
It just has hydrogen as an R group.
So there's no chiral.
Okay, so there's no chiral center. We have those two hydrogens instead of an extra carbon chain or something else. Isoleucine and threonine contain a second stereocenter. The natural question that would come out of that would then be, what's the stereocenter? RS. I won't ask that, but that just happened to be my natural response off of that. There's no point in then making you go through that again. Your uncommon amino acids, there are a few of them out there. They're usually derived from the 20 basic ones. So you're looking at structural effects, structural changes to them. Sometimes it involves just adding an extra hydroxy group to them. Why would we have some of these uncommon ones? We want to change the functionality around mildly, so we might put on an OH. What is that going to do? It's going to change. It shouldn't affect your isoelectric point. Why not? An OH, you're looking at typically an alcohol. Your alcohols aren't really acidic or basic. What will it change? Changes the structure a little bit, goes back to polarity. OH is a very, very polar bond. We can turn a functional group that is typically nonpolar to now being polar. So it could change its general location in the protein, which hopefully we'll get to. Yeah.
Isn't tyrosine derived
Tyrosine from phenylalanine.
Well, tyrosine just has an OH, the same structure. Like you said.
Oh yeah. They both must show up frequently enough. So common versus uncommon is, again, probably an arbitrary line. So both phenylalanine and tyrosine probably show up fairly frequently. So while they are probably derivatives of each other, since they're both there in high enough concentrations, we call them both common. pKa's of your carboxyl and your amino. You should know those. Questions on those? Okay. I think there's some little bit more stuff. We're looking at peptide bonds. So now we can combine the amino acids. They're going to be combined through peptide bonds, which is our biochemistry term. Since we're all chemists, we actually know better, and it's actually the amide bond. In the process of doing that, what happened? Okay, so we refer to it as our amide bonds. Let's write that in here. Should have picked a better picture. I should have drawn my own picture. Actually, we can kind of get away with this one. What type of chemical reaction is this? Dehydration works. Dehydration. The primary one that I was going for there was a condensation. Now we could actually tweak that a little bit more, because condensation reactions or even dehydration are typically in reference to one reaction. When we're looking at amino acids, it's not one reaction, it's multiple reactions. So it's not just a dehydration or a condensation, it's a condensation polymerization. We're actually forming polymers in the process of this. Okay. Why is it a condensation? You're losing water from within that structure. Okay. In general, we'll jump to the very end on that. Note your kind of standard notation. You almost always start amino on the left, acid on the right. So if you're given an amino acid chain and they list out a whole bunch of single letters,
Yeah.
okay, so we've got, did I actually pick,
No, it was ED.
but one, did I pick one that actually worked? Hopefully I did. Okay, if you're asked to figure out the charge on this amino acid, to do the charge, what do we need to know? We need to know the structure, so we need to know the R groups, but is there something else? We need to know the pH, but then we also have to be aware of an amino acid. Just because we've polymerized out doesn't mean we've lost the amino on one side or the acid on the other. We still have the amino functionality hanging off our E group, and we'll still have the acid functionality hanging off our T. Okay. To decide charges, you need to know the pH to decide should hydrogens be on those or not. Okay. To have the COOH, what do you think our pH is?
Big.
It's got to be below 2. We're probably looking at something like 1. We don't want to go high because remember we're trying to force hydrogens onto the structure. So we have to have a huge concentration of hydrogen ions, so low pH. To have NH2, that's really what you were thinking. We want to be above 9 because we're trying to remove hydrogen from the structure, and that's why it's NH2 as opposed to NH3. Okay. So as I've got it drawn, would this structure ever exist? No, because I can't put two drastically different pHs. Okay. Any interesting features about our peptide bond? Or, sorry, let me clarify that, our amide bond. So I'm going to erase that middle section. And I want to draw in an example amide bond. Yes. Why is that resonance structure important? So it does a couple things for us. Number one, that resonance turns this amide bond into effectively a double bond, which means we minimize a lot of the rotation about that. Okay? So that kind of reduces the amount of motion that we could expect in our larger structures.
It also lowers the energy barrier for formation.
Does that as well. We lower the energy barrier for that reaction. I wouldn't worry about that as much, but it's true. When we look, remember all the stuff we've been looking at is all this acid-base characteristics. This nitrogen out here can pick up a hydrogen. Well, this nitrogen has a lone pair. Can it pick up a hydrogen? No. Why not? It's involved in the resonance. The basicity of this nitrogen is actually pretty basic. It can pick up hydrogens at will. This one, being embedded in the structure and embedded with resonance, has lost that basic characteristic. So when you look at the amide bond, it's not basic. Okay? It won't react as a proton acceptor. Okay? Or donor, for that matter. Okay? Is that a question? Yeah.
Yeah, I was just gonna say, one of the exercises, it was pretty much asking the overall charge.
Mm-hmm.
And I mean, that's why those become negligible.
It's not that they're negligible, they don't react. So, yeah, those won't act as bases because of this resonance contribution.
So on the end and
The
R.
end and the R groups, that's it. Okay, when trying to evaluate charge, which is ultimately looking at, again, your acid-base characteristics. All right. Other questions? Okay. So intermolecular forces plays a huge role in Chapter 4 again. So remember the interplay of these. First off, when we're looking at our proteins, typically where are they found? What solvent dissolves our proteins? So we're really looking at how water is going to contribute to the overall structure of our protein, okay, or our polypeptides. So water is going to give us what intermolecular force? Hydrogen bonding. So if we're going to look at our protein, we're probably going to look at a lot of hydrogen bonding amino acids on the outside of the protein so that they can interact with water. And on the inside, we would expect hydrophobic. Okay, so they're repelling away from the water. Okay, so that's a really important thing to realize. You will definitely see a question on that because I just said it. So that will show up on your exam. And it's also a good way for me to go through and also test your knowledge on your amino acids. So the question that I could ask is, of the 5 amino acids that you memorized, which ones would be found on the inside of the protein? List the structure, 3-letter code, 1-letter code. Right. So valine is one, or valine, I don't know. Right. Would be one that you would expect to find on the inside. I could also just as easily ask it which ones you find on the outside. So if you went back to those categories that we talked about with our amino acids in chapter 3, one group, I think it was you guys, put together hydrophobic and hydrophilic, and then separated the hydrophilic out into separate subcategories. Okay? That's a reasonable comparison, because when we're looking at protein folding, we're concerned about hydrophobic and hydrophilic interactions. Okay? So keep that in the back of your head when you're going through and trying to memorize amino acids. Should not be all of the exact same type, because we want to see a little bit of variability. So our individual protein divisions, mainly because I had these images already. Our leftmost structure there is ATP synthase. What's happening in the middle? Anybody know what's happened there? Still ATP synthase. This section here is just this top. This section down here is just this bottom part. Okay? So all of these different colors are actually representing what? Different proteins. ATP synthase is actually a massive protein made up of a whole bunch of smaller polypeptides, okay, or miniature proteins that have now self-assembled into this much larger structure. So what I'm kind of doing is breaking down from this structure down into smaller subcategories, and then down even smaller. This sequence on that far right is actually a single polypeptide out of our green section here. The green section is a combination of anywhere between 10 to 12 of those subunits. Okay? So it's kind of neat that these can all self-assemble out. So what we're trying to represent is kind of a breakdown in the overall structure. The ATP synthase fully put together, we're looking at what's referred to as our quaternary structure, which is the assembly of multiple independent proteins. We're still kind of looking at our quaternary in that breakdown, but we'll call it quaternary / tertiary, because what we're looking at is how each of those individual proteins fold. Okay, so in our last image, we're looking at our tertiary / secondary. It's going to be our tertiary because that's the completely folded protein, but it's also our secondary in this case because we're looking at how the protein itself folded up into smaller structural motifs. That structural motif in this case happens to be a helix. Okay. Can we break it down even further? Yeah, we can go all the way to primary structure. Primary structure is now looking at just the sequence of amino acids. A lot of data has been generated. Oh, that's why. Gotta close that. Whoa, don't close that. A lot of data has been generated and collected. Let's see, is that working? And PDB is the protein data bank. And so every time someone goes through and isolates a new protein and determines some structural information, they can go through and post their information to PDB. And what we see, oh, come on. What we see in this case, okay, is now the sequence for that C subunit, okay, from, forget which site or which source it came from. But you see your one-letter code for each of your amino acids. What we're looking at there is the primary structure. They then add some more information and said within that primary structure, we also have some secondary structure. Okay, those of you that had some background, what's the arrow represent here? Okay, maybe you don't have that background. The arrow represents looking at a beta sheet. So that sequence of amino acids is generating a strong interaction with another potential sequence. Okay, another arrow. The squiggly lines are alpha helix. So the amino acids within that section are generating that helix shape. Okay? So there's a lot of information out there for individual proteins and what their overall structures look like. Yeah? Which loops? Oh, the little bumps? Not positive on that. I'm going to guess they're turns in the protein. Can I come back? Cool, I think it did actually work. It did come back. So what we're going to be concerned about is how we can kind of look at each of these individual subcategories and what information is found there. So our primary structure is just the sequence of our amino acids within our peptide chain. Okay? All of these different categories are related to each other, and so we do need to be aware of those relationships, but our primary is ultimately just the sequence of amino acids. Okay? And that sequence is really, really important to the overall structure. So if we take that big long chain that we were looking at before and we change one of those amino acids, we can end up changing that overall structure drastically. Okay? The example given, whoops, okay, never mind. We'll almost get to that example. So this is where you would need to know what all of your individual side chains are. So we've got another list of, or another way to list out the amino acids. You'd have to know what physical properties they're going to generate, how they're going to interact with other things. Your hydrophobic, your hydrophilic, your acidic, basic. So it's one of the reasons why typically you're required to memorize all of the amino acids, so that when we start talking about more advanced structures, you can start to decide what things should show up where and how they apply. All right? So how important is that exact, oh, did I lose connection? Oh. Now that I skip slides. Okay. How important is that overall sequence? If we take a look at red blood cells, we've got normal red blood cells and sickle cell. Okay. Pretty different overall structures. Okay. Sickled red blood cells, not really a good thing to have. Okay. The odd shape can cause blockages and they have issues picking up oxygen as cleanly. What they've gone through and found is one of the primary causes between a healthy red blood cell and sickled cells comes down to a single amino acid. What they're looking at is the sequence of amino acids, and what they're labeling as 6. That 6th amino acid used to be glutamate. In the sickle cell, it's been changed to valine. That single change has disrupted the structure of that protein so much that it now disrupts the structure of the overall cell drastically, just based on a single amino acid. Okay. That's a pretty massive overall change. Why would it have such a big change in the overall structure?
Because it won't form correctly.
Okay, why?
Because the interaction would be different. Maybe hydrophobic.
There we go. You'd have to know what the structure was of your glutamic acid or your glutamate and your valine to see what the difference in the intermolecular forces are. So if we pulled open the structures, sorry, go ahead.
Isn't it acidic?
Your glutamic acid has an acidic functionality, okay? Very high polar, very hydrogen bond capable. Valine, extremely hydrophobic, okay? So within that sequence of chains, okay, the rest of the protein may actually fold normally, but where the active site is within that protein, where it's actually performing its duties, the amino acid is no longer there to actually allow for that proper interaction either with oxygen or maybe with another protein. And that then prevents the blood cell from doing its job appropriately. Okay? So your primary amino acid sequence can be very, very important. And that's where we'll talk, or where you'll see, can expect more questions going all the way back to chapter 3 and saying, do you remember what those individual structures were? How do those changes affect the overall results? Okay? In this case, we're looking at a really drastic change in intermolecular forces, but it could have been something much more subtle. We could have gone from, testing my knowledge, alanine to valine. What was alanine's structure? Oh, I wanted glycine. Never mind, I remember. Glycine. What's glycine? Just hydrogens. If we switch from glycine to valine, would that be a big difference? Both of them are relatively hydrophobic, so why would we expect any big changes? This chain is substantially larger. perhaps where the glycine was located was in a position that was sterically very, very small and hindered. So the glycine didn't affect the overall folding. If we put in valine now, is that valine? Yeah, never mind. Okay. With valine now being sterically much larger, that can prevent the proper folding further on down the line. Okay. So it's a very dependent process on structure and intermolecular forces. So it's not purely just positives and negatives, hydrophobic and hydrophilic. We also see a lot of steric effects, okay, and potentially even resonance effects, which should sound familiar. What are the three primary contributors to organic chemistry? Resonance, sterics, and ultimately electronegativity, or your polarity. Okay? So everything that's picked up in organic chemistry is really playing an important role in biochem as far as how these things interact and propagate. I got a little more time. Okay. So secondary structure. What's happening with secondary structure? I got a minute. Secondary structure is now how does that primary structure, the sequence of amino acids, interact to generate now larger structures. So instead of now looking at just the side chains, but how can the protein, that sequence, change its overall conformation. So we're looking at very small changes to now looking at how that whole protein can start to interact with other things. Our secondary structure is primarily due to the backbone within our protein. So we're looking at the amide linkages through the backbone. Okay? So we're less concerned about our side groups when we're looking at secondary structure. Okay? What do we see in the backbone? So this picture gives us some hints as what could possibly show up. Okay? The first thing that most people would notice, which is what I noticed too, we have a nitrogen-hydrogen bond, which means we have the capability for hydrogen bonding. If we look at the next sequence, we've got a carbon-oxygen, very polar bond. That oxygen can act as a hydrogen bond acceptor. Fine, I already started it. A hydrogen bonder acceptor. Oh man. And a hydrogen bond.
Don't.
Maybe I should just stop. And a hydrogen bond donor. Should have quit while I was ahead. So we've got the hydrogen bonding ability. Those hydrogen bonds within the amide bond background can start to interact with other side chains, and it can also interact with protein or amino acids further down the chain if we get proper bond angles. So remember, we said the amide bond can't rotate, but around our R group carbon, we can get free rotation. Okay, depending on primary structure. Where would the primary structure prevent that free rotation? Would we see a double bond coming from our group? No.
Doesn't it have to do with the,
Sterically hindered might do it. Go ahead.
like the negative on the oxygens, those would be repelling against each other, wouldn't they?
You're still looking at something a little bit distant there. We could potentially see that. When is that bond rotation absolutely prevented? P? Proline. What's the issue with proline? It's bound with the backbone. Okay? So again, it goes back to the structure of those amino acids, and I'm definitely stopping there. So as far as exam 4 goes, or exam 1, we'll get up to the basics of secondary structure, but we won't talk, or you won't see questions on alpha helices or beta sheets or any of that. Okay? So it'll be just kind of on this slide. I will go through homework 4, hopefully right after class, and sort it out and try and split it into 2 assignments.
Mainly the first 3 chapters, really?
So it's really just going to be the first 3 chapters, and then you need to be aware of primary structure and ultimately what contributes to secondary structure. Everybody heard that? What contributes to secondary structure? It's the backbone of your peptide. We're not looking at the side chains yet.
Okay.